This video is going to review mainly transcription and translation of DNA to make proteins. So one of the very first things we need to understand is what a gene is. A gene is a piece of the chromosome or a piece of the DNA, a segment of the DNA that will code for a specific protein. So humans have about, they're saying 19,000 genes. That number is a little unclear and it could change, but 19,000 seems to be the most recent update. And the entire human genome is about 3 billion base pairs long. One gene, one segment of DNA that codes for DNA, can range anywhere from 1,000 base pairs long to 2.4 million base pairs long, and that's the longest one that they have found so far. And you can see here, I've put up an example where it shows the genes on chromosome 1. Chromosome 1 is 246 million base pairs long, and you can see the extensive list of genes that can be found on chromosome 1. So a gene is a segment of DNA that codes for a protein. You need to understand that there are 3 types of RNA. There's mRNA, rRNA, and tRNA. The M in mRNA stands for messenger. Okay, the purpose of this is to basically make a copy of the DNA. Remember, the DNA can't leave the nucleus, and mRNA sends the message of what's in the DNA to the ribosome so that a protein can be made. rRNA is structural. It makes up part of the ribosome. It doesn't really do anything per se, but it is structural. It makes up part of the ribosome. tRNA is important because tRNA has the anticodon, which carries the correct amino acid to the ribosome to make the polypeptide chain. So transcription is basically where the DNA is copied into an mRNA molecule. So again, the DNA cannot leave the nucleus. So in order to get the information in the DNA to the ribosomes, which are outside of the nucleus, a copy of the DNA needs to be made. And the cell does this using mRNA. The mRNA then leaves the nucleus and goes to the ribosome. And the ribosome will begin reading the mRNA. So transcription is just copying the DNA into a molecule of mRNA. So again, the mRNA then leaves the nucleus and goes to the ribosome. Now at the ribosome, so here we have the ribosome. This is the mRNA. At the ribosome, the mRNA is being read and translated. Hence, this portion of protein synthesis is called translation. So the ribosome reads the mRNA in groups of 3, and those groups of 3 are called codons. And the tRNA has the anticodon, so the opposite of the codon. And that basically tells the tRNA which amino acid needs to go next in the amino acid sequence in the polypeptide. So remember again, the ultimate goal here is to make a protein. Once all of these amino acids are linked together, the protein folds and makes a specific shape, and then it can perform its function. This is a really, really great review video. I really strongly recommend that you watch it, and it will I think, really clear things up for you in terms of transcription and translation protein synthesis. Something important that I'd like you to know is the path protein synthesis takes in the cell. So protein synthesis starts in the nucleus with transcription, where it's making a copy of the DNA to mRNA. It leaves the nucleus through a nuclear pore, and it goes to a ribosome. Now, there are ribosomes out in the cytoplasm, but there are also ribosomes attached to the rough endoplasmic reticulum. Remember, that's what makes the endoplasmic reticulum look rough. It has a bunch of ribosomes attached to it. So the ribosome makes the protein. Now the protein travels through the rough endoplasmic reticulum to the smooth endoplasmic reticulum, where it's packaged into a vesicle. It's then taken to the Golgi apparatus, where it's finalized, whatever happens to it, and then it's packaged again into another vesicle, and a vesicle that kind of sends it out into the body is called a secretory vesicle. So then it goes to the cell membrane and fuses with the cell membrane to release those proteins into the body. Some proteins will stay in the cell. So you need to know how to do transcription and translation. So I'm going to do a little practice here. So remember, the rule for RNA is that there is no thymine. It's uracil. So it's still, T still is bonded to A, but instead of A binding to T in the mRNA, it binds to a U, and then C still binds to G. Then we have here, we'll get C, G, C. C will bind to G, G again, and then A will bind to T. So we can have A in mRNA. But here, in DNA, where it would have been a thymine, it's a uracil. And C's bind to G. Here we have C, C, C. And then A, C, C. So that's transcription. Just copied the DNA into an mRNA molecule. Now this codon chart is very important. It will help you determine which amino acids correspond to which codons. So we have the codon here, A, U, G. To read this, we go from the middle, which is the first letter, so we have A, and then we work our way out, U, G. So the first amino acid in this protein is going to be methionine. And I typically abbreviate, and that's fine for you to do as well. Our next one is CGC. So working from the middle out, CGC gives us arginine. Then we have GGA. Working from the middle out, GGA gives us glycine. UCC gives us serine. CCC gives us proline. And ACC gives us threonine. And now you have translated the mRNA molecule to get the sequence of amino acids, which will build the protein. In this example, it doesn't only ask for mRNA, it also asks for tRNA. So tRNA, again, just provides the anticodon to the mRNA. You don't really need it in order to do translation, but I'm going to do it here for you just to show you. I think that's CUA. I'll go back and fix it if not. Okay, so the rule basically here for tRNA is that if there's an A, it will bind to a U, and if there's a C, it will bind to a G. So again, you're just replacing the uracil, you're replacing the thymine with uracil. No T. Now, when you go to figure out the amino acid sequence, when there's tRNA present, you are completely ignoring the tRNA. You are still only using the mRNA codon to find the sequence. And here I've provided a different example of a codon chart, just in case you see this type in the future. So here we read what's on the left first. So if our first letter is A, I'm going to find A, which is here. Second letter is also A, so second letter is right here where the two cross. And then G, I need to find the third letter over here. So where the three letters cross each other, that's going to be the answer. So in this case, we have lysine. Then CUA, C, U, and A will be leucine. A, U, C will be isoleucine. UAC will be tyrosine. GGC will be glycine. And UUC will be phenylalanine. This one is a little bit more challenging. I'm going to let you try this one on your own. So pause the video and give it a try. And we'll go over the answer to this in class.